Hello, Bruins. This is a continuation of cellular respiration screencast, and we're going to talk about glycolysis. So, as I was telling you, glycolysis, this is my flow chart, my graphic organizer to remember the way glycolysis works. So, again, glycolysis takes place in the cytoplasm, all right? So, it occurs in the cytoplasm, and no oxygen is needed. Okay, and what's the purpose of glycolysis? So the purpose of glycolysis is basically to split the sugar in half. So you have your, say, 6-carbon sugar, and you're going to split it into a 3-carbon sugar and releasing some energy in form of ATP and any electrons picked up by electron carrier NAD plus, which converts to NADH. Okay. And so when you're studying these reactions, cellular respiration, kind of concentrate the purpose, what do you need, your reactants, and basically you need glucose, you need NAD plus, and to get that process started, we need to input a little bit of energy, so we need 2 ATPs, and we need 4 ADPs to convert it to ATP. And what do we get at the end? Our products is pyruvic acid, and ATP and NADH. So here's a graphic organizer to kind of help us remember. So we start with glucose, 6-carbon sugar, right? And this is not just one reaction, these are a series of reactions, and just to simplify things, so we input 2 ATPs, right, and our net gain, and then we get 4 ATPs, right, and so our net gain is basically 2 ATPs at the end. So we get 2 ATPs, and when we break those bonds, we pick up those electrons, and NAD plus, our high energy electron carrier, is reduced to NADH. And basically at the end we get this 2,3-carbon compound called pyruvic acid or pyruvate, and that's basically glycolysis. Now if there's oxygen, then the 3-carbon molecule pyruvic acid will go into the mitochondria. If we don't have oxygen, they'll go through fermentation, and depending what kind of organism you are, there are 2 types of fermentation. So the purpose of fermentation is to keep glycolysis going. And to keep glycolysis going, you got to regenerate NAD plus to keep glycolysis going. Okay, so here this part is basically glycolysis. So you got your glucose, and when you break that 6-carbon sugar, you pick up some electrons by NAD plus and becomes NADH, and you make a little bit of ATP, and you make your 3-carbon molecule called pyruvic acid or pyruvate, same thing. Now there are 2 types. One type is the alcoholic fermentation, so this one shows you your alcoholic fermentation, and this one is our lactic acid fermentation. Okay, our alcoholic fermentation will release that 1 carbon as carbon dioxide, and electron is used from NADH to make NAD plus, and it becomes a 2-carbon alcohol called ethanol. Okay, now lactic acid, it remains a 3-carbon molecule, but it'll take up that electron and become lactic acid. And depending on what kind of organism you are, usually yeast and some bacteria will do alcoholic fermentation. And lactic acid are done by animals. If you ever felt that burn after you do exercise, that's lactic acid being formed in your muscles. And some bacteria like to make yogurt, sour cream, kimchi, sauerkraut, all of that is doing lactic acid fermentation.